The outline for this chapter is we're going to start off by talking about amino acids. These are the building blocks for proteins. Proteins are considered polymers. Amino acids are considered monomers. We're then going to put some of those monomers together to form small molecules we'll call peptides. And then we're going to put those monomers together into larger molecules, which we call proteins. And then we'll discuss some of the characteristics of those proteins. A protein is a naturally occurring, meaning it's made by living organisms. It's an unbranched polymer in which the monomer units are amino acids. This is the generic formula for an amino acid. It contains an amino group and a carboxylic acid group on the same molecule. This is a representation of the protein polymer chain. Each one of these units here represents an amino acid that has polymerized to form this long continuous chain, which is made up of polyamide units. Proteins are the most abundant molecules in cells after water. That's about 15% of a cell's overall mass. Proteins are largely composed of carbon, hydrogen, nitrogen, oxygen, and some sulfur. They also often contain a little bit of iron and phosphorus. And we'll look at some of those specialized amino acids and proteins further on in the chapter. So let's look at how those monomer units have come together. If I look at a carboxylic acid, it can react with an amine to form an amide bond right there. And if I look at this polymer chain here, here's a whole bunch of amides. So if I take a carboxylic acid, shown here on this amino acid, and I react it with an amine, also shown here on this amino acid, they can react to form amides or peptide bonds. Let's take a look at those monomer units and their abundance and their structure. An amino acid is an organic compound that contains an amino group, shown here in blue, a carboxylic acid group, shown here in green, an R group, which is often known as a side chain, in which all of these functional groups are attached to the same carbon, which we're going to call the alpha carbon here. If all that is true, that is an amino acid. Even though there are many different amino acids, it's only the R group that determines the difference between different amino acids. There are over 700 known amino acids. However, there are only 20 standard amino acids that are present in most living organisms. In other words, there are just 20 different R groups. So in every one of these 20 different amino acids, they have the same amino group, they have the same carboxyl group, they have the same alpha carbon. The only difference between those 20 is that they contain a different R group. Also note that the alpha carbon is a chiral carbon. It has 4 different things attached to it, which implies that it has stereochemistry. Let's now look at those 20 different common amino acids, and we're going to group them into 4 different groups based on their physical and chemical properties, and those are again going to be determined by the R groups that are attached to the alpha carbon. The first group is nonpolar amino acid. That means that they contain a hydrophobic side chain, a hydrophobic R value, like just carbons. And there are 9 of these. One of them is glycine, which contains hydrogen as its R group, alanine, which contains a methyl group, valine, which contains an isopropyl group, leucine, which contains an isobutyl group, and isoleucine, which contains a sec-butyl group as its R side chain. Those are all hydrophobic, they're nonpolar. There are 4 other, One is this proline molecule, which is actually cyclized back to the amino group here. I have phenylalanine, which has an aromatic ring bridged with a carbon to my alpha carbon. I have methionine, which has a sulfur in there. It has 3 carbons and a sulfur. And I have tryptophan, which has these 2 bridge cyclic compounds, both of which are aromatic. Those are going to be nonpolar amino acids. The second grouping of amino acids are the ones that are polar, neutral amino acids. Polar meaning that they have some distribution of charge. Neutral means that they're not ionic. That also indicates that they're either non-acidic or non-basic. We have 6 amino acids that belong in this category. Serine, which contains a hydroxyl group, which is polar, but it's neutral. I have cysteine, which contains a sulfhydryl group, which again is polar but neutral. And I have another amino acid called threonine, which has a hydroxyl group on it, but 2 carbons as compared to serine. I have asparagine, which has both a carbonyl group and an amino group on it. I have glutamine, which also has a carbonyl group and an amino group on it, so these would be amides here. And finally, I have tyrosine, which contains an aromatic phenol compound out here. These are all polar, neutral amino acids. The last 2 categories are polar acidic amino acids. They typically contain a carboxylic acid group for their side chain. One of them is aspartic acid, and another one is glutamic acid. Notice they both have a carboxylic acid group out here, one separated from my alpha carbon by 1 methylene group, and the other one separated to my alpha carbon by 2 methylene groups. Polar basic amino acids typically contain a nitrogen atom in that side chain group, which would be basic. It has a lone pair that's available to pick up a hydrogen. In this case, in histidine, this nitrogen here is basic. This one is not. If I look at the amino acid lysine, it has an amino group out here with a lone pair of electrons. It's a basic amino acid. If I look out here, I can see that I have several different nitrogens out here, some of which have lone pairs out here. For example, this one, and this one. These are all polar basic amino acids. This last one here is arginine. Those are our 20 amino acids that are standard amino acids, meaning they're common in life. Notice that when we describe those and name those, we used common names for amino acids. That comes historically again because the IUPAC name for these molecules can be complex. So if we look down the list here of alanine, arginine, aspartic acid, asparagine, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, thionine, tryptophan, tyrosine, and valine. Those are our 20 amino acids. Even naming them is often too long when we want to write down which amino acid we're talking about. So we often use these sort of abbreviations for amino acids. Most of them are just the first 3 letters of the amino acid name. For example, alanine, its abbreviation would be ALA. Arginine, its abbreviation would be ARG, and so forth, except for 3 of them. For asparagine, it is abbreviated ASN. For glutamine, it's abbreviated GLN, and for tryptophan, it's abbreviated TRP. That's because some of these would have the same names if they were named just by the first 3 letters. For example, let's look at glutamine. I have glutamine, which is GLN, and I have glutamic acid, which is GLU. Both of them would be GLU, so they just changed a few of them to make it easy to differentiate. We will see later on that we actually then go to one-letter symbols to describe amino acids when we start talking about them in the context of proteins or peptides, those polymer chains that are made out of my amino acids. In this molecule, this is just the representation of phenylalanine, PHE, leucine, LEU, serine, SER, cysteine, CYS. Let's do a practice exercise where we identify the alpha carbon in these three molecules, these three amino acids. Let's classify each of them based on their side chain polarity, and then let's actually name them. And here are our choices for naming these three different amino acids. First of all, let's look at the first one here. What I'd like to do first in finding the alpha carbon is to look for the amino group, look for the carboxylic acid group, and the carbon that bridges them will be my alpha carbon. If I look at the side chain here, notice that it has a nitrogen out here, and this one here is going to be a polar neutral because this is an amide, and amides are neutral molecules. They're not acidic at all, or they're not basic. If we looked at this molecule, I might have to go back to the table. This is glutamine. If we look at this amino acid, I'm going to identify my amino group, I'm going to identify my carboxylic acid group, and then the carbon that's attached to both of them would be my alpha carbon. That means the rest of this molecule is my side chain. If I look at this side chain, it's only made up of carbons and hydrogens. That means it's going to be a nonpolar group, and this one would be isoleucine. If I look at this molecule right here, again, identify my carboxylic acid and my amino group. The carbon which bridges both of those two groups is my alpha carbon. I look at my side chain here. This carbon here has a lone pair of electrons on it that are free to act as a base. This one here would be a polar basic amino acid, and this one happens to be histidine. There are a number of amino acids that we must obtain from dietary sources. We call these essential amino acids, much like we talked about fatty acids, which we need from dietary sources called essential fatty acids. In general, adequate amounts of some amino acids are not biosynthesized by our bodies. That's usually because they're very expensive energetically. In other words, it's a complex process that takes a lot of energy. 10 of the 20 standard amino acids are considered essential, meaning we need to get them from dietary sources. Arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Notice there's an asterisk here by arginine. That's because typically arginine is required for growth in children, but it is not an essential amino acid for adults, so it's got some asterisks beside it. In fact, I can go to the drugstore and actually buy supplements, in other words, pills that contain essential amino acids. There are some nutritional scientists that actually discuss these essential amino acids slightly differently than the last slide. If we look at the non-essential amino acids, those are the ones that our bodies make plenty of, and those include alanine, asparagine, aspartic acid, and glutamic acid. There is a group that is called essential amino acids, and there are 9 of them listed here, just like on the last slide, with glutamine here listed as a conditional amino acid, and there are some other conditional amino acids. And when we talk about conditional, we mean I might need more of this amino acid when there's times of stress on the body. For example, if I'm ill or if I'm injured, I might need more of one of these amino acids, so it becomes essential during times of stress or illness. Let's look at some of the physical properties of amino acids. Pure amino acids are white crystalline solids. In other words, if I have several different amino acids next to each other, I could not tell which one it is based on just looking at it. Amino acids often decompose before they melt. So an example of decomposing is if I were to burn an amino acid, like in this steak here, it would start to burn rather than melt and decomposes. And they are not very soluble in water. So if I look at amino acid and put it in water, it tends to just fall to the bottom, precipitate out as a crystalline here. They all undergo chemical reactions either during cooking or in the body. Those properties of amino acid are determined by the basic group, the amino group, the acidic group, the carboxylic acid, or the properties of the side chain R group. If one looks at the generic structure for an amino acid, the alpha carbon here has an amine group attached to it. It has a carboxyl group attached to it. It has a hydrogen attached to it. And it has an R group attached to it. So 19 out of the 20 common amino acids have 4 different things attached to it. Only glycine, where R group is equal to H, does not. So that makes those 19 common amino acids chiral molecules. Okay. If I have a chiral molecule, there exists a left-handed version and a right-handed version of that. In other words, enantiomers of each other. Most amino acids found in nature are the L isomers. Also recall that naturally occurring monosaccharides typically are the D stereoisomers. There are a few bacteria that do have D amino acids. Let's just look at sort of the different enantiomers, the L amino acid versus the D amino acid, and look how we name them. If I sort of draw them with the carboxylic acid group at the top and the R group at the bottom, if my amino group is on the left-hand side, that's the L amino acid. If my amino group is on the right-hand side, that would be the D amino acid. So in general, this sort of looks just like a Fisher projection, and that is actually how it's drawn. I put my long carbon chain in a vertical axis, starting with the most oxidized group at the top, going down to the least oxidized group, in other words, the carboxylic acid and the R group. If the amino acid group is on the left, L amino acid. If the amino group is on the right, it's the D amino acid. Let's do a practice exercise where I've drawn the Fisher projections of 3 different amino acids. I've put the carboxylic acid group at the top and the R group down at the bottom. Let's identify whether the amino acid is the L or the D stereoisomer. If I look at this first one, here's my alpha carbon as drawn in the Fisher projection. My amine group is on the left-hand side. That would make this the L stereoisomer. In the second molecule, drawn in the Fisher projection, my amine group is on the right-hand side. That means that would be the D stereoisomer. And also, if I look at this third example here, here's my alpha carbon. Here's my amine group is on the right-hand side. Therefore, this is the D stereoisomer. Let's now take a look at the chemical reactivity of amino acids, and let's start off by looking at the acid-base characteristics of an amino acid. If I look at the generic structure for amino acid, I have my alpha carbon here in the middle. I have an amine group shown here in blue. I have a carboxylic acid group shown here in green. I have my R group and I have my hydrogen. The group on the left is basic. That means I have a nitrogen with a lone pair of electrons. It would like to react with some acidic component in solution to have another hydrogen put on it because it's basic. My carboxylic acid functional group here, it's acidic. It would like to give up one of these hydrogens here to something else in solution. So this molecule can undergo both acid and base chemistry at the same time. And that's exactly what happens under physiological conditions, meaning in a body. My amino acid here, my carboxylic acid group wants to give up a hydrogen. My nitrogen wants to take on a hydrogen. So there's an equilibrium between the uncharged structure and the charged structure. Note that this molecule is still overall neutral. I have a plus on the nitrogen because it has an extra hydrogen on it. I have a negative charge on one of the oxygens because it's given up its hydrogen. We call this structure a Zwitter ion. A Zwitter ion is a neutral molecule where that molecule has atoms that bear a positive charge and a negative charge on it. So the carboxylic acid group donates a proton to become negatively charged. The amino group accepts a proton to become positively charged. We can also look at that as a distribution of charges here. My nitrogen contains my negative charge, where my negative charge is distributed between both of those oxygens. Both of them tend to be equal. So this is the Zwitter ion form of an amino acid, and that's what an amino acid looks like under physiological conditions. That means in the body. One can think about that Zwitter ion in a body. If I am in a neutral solution in the body, my amino acid acts as a Zwitter ion. It's got a positive charge and a negative charge. If I then move that amino acid to a part of the body that's very acidic, I would then reprotonate my carboxylic acid to form this acidic solution where I have a positive charge on my nitrogen and I have a neutral charge on my carboxylic acid. If instead I go to a basic part of the body where I have lots of base in it, I'm going to pull that hydrogen off of my amino group and I'm going to leave the hydrogen off of my carboxylate group, and so I'm going to have a negatively charged amino acid. One can think of this as sort of like a titration. Here I start out at pH near 0, which means I'm in very acidic conditions. So as I start to add sodium hydroxide, for example, to my solution, which contains an amino acid, all of my amino acids look like this positively charged species. As I get over here to where I've added an equivalent of sodium hydroxide, I actually reach a neutral solution, pH 7 here. That is the isoelectric point of my pH titration curve here, and all of my molecules exist as the neutral Zwitter ion. As I keep on adding base to bring my pH up to near 13, I see that I have all my species exist as this negatively charged carboxylate amino acid. And different amino acids have different isoelectric points, meaning the pH at which they are neutral in solution and exist as the Zwitter ion. Let's look at some of the pH values for some of the solutions of the human body. If I look in the stomach, I notice that our pHs are very acidic. Therefore, my amino acid will exist as the cation. As I look at areas which are near neutral pH around 7, my amino acids will exist as the Zwitter ion. If I look at some areas, like in the pancreas, they are basic in nature, and those will exist as the anion. Remember that some amino acid side chains are also ionizable. In other words, they can function as both an acid or base. Let's look at one of those molecules here. For example, if I look at this amino acid, I have the base amino group and my carboxylic acid group. I have a secondary carboxylic acid group on my side chain. In this case, if I'm at low pH, all three of these functional groups are protonated. As I start to increase the pH, like in moving to a different part of the body or doing a base titration, I start to remove the most acidic hydrogen, That would be this one here. So I now form what we look at is a Zwitter ion here. I have one positive charge and one negative charge. That's a neutral molecule. If I move to another part of the body, which is more basic, I pull off the next acidic hydrogen, which would be on this side group carboxylic acid here, to form the net negative 1 charge on this molecule. If I continue to increase the pH or move to a more basic part of the body, I then remove my last acidic hydrogen that came from this one to form this net -2 net charge amino acid group. It all depends on what the pH of the compound is what the charge looks like in amino acid. In neutral solutions, I have the Zwitter ion. In acidic solutions, I have a positively charged ion. In basic solutions, I have negatively charged ions. Let's now do a practice exercise where we draw the possible ionized forms of the amino acid histidine. This is actually a structural diagram of that. I have my alpha carbon, I have my carboxylic acid group, I have my amino group, and this is my side chain. Let's first identify the acidic and basic groups. Up here, my hydrogen on my carboxylic acid is acidic. My lone pair of electrons on my amino group is basic. This lone pair of electrons on my amino group is basic. However, this lone pair on the amino group is not basic. It's tied up in these alternating carbon-nitrogen double bonds, which makes it aromatic. So it's not basic at all. So let's look. If I'm in a very acidic situation, I want to protonate my carboxylic acid. I want to protonate my amino group, and I want to protonate my second amino group. So I draw the diagram here. My carboxylic acid, I have a cation that exists. It's a +2 charged molecules. As I start to increase the pH, I start to remove the most acidic proton, which would be in this carboxylic acid. In this case, I have a negative charge now. I have a carboxylate ion up there. I still have 2 positive charges on my amino groups. If I start to increase the pH again, making it more basic, I remove the next most acidic hydrogen, which happens to be this one down here. That was determined by experimental process. They determined which of these 2 nitrogens here was more acidic. And if I increase, now that's my Zwitter ion now. If I increase the pH more, I remove that final hydrogen on that amino group to form the anionic species of my amino acid. Cysteine is a unique amino acid in our group of 20 standard amino acids in that it contains a sulfhydryl group, an SH, often referred to as a thiol group. That sulfhydryl group imparts cysteine with some special chemical properties. If I look at the chemical reaction between 2 sulfhydryl groups, if I oxidize them, I form a disulfide bond. If I break that disulfide bond, I reduce them to form my thiols again. This is a reversible process. So if I look at cysteine, if it is close to another cysteine molecule in the right conditions, I can oxidize those 2 sulfhydryl groups to form a disulfide bond between those 2 cysteine amino acids. I've actually done some unique chemistry. Let's now start talking about peptides, which are the precursors for making long polymer proteins. Under proper conditions, amino acids can actually bond together to form small amino acid chains, which we refer to as peptides. If you recall, I have an amino acid which contains a carboxylic acid group and amino group. that carboxylic acid group can react with the amino group of a second amino acid, forming a amide bond, sometimes referred to as an amide linkage, or when we're talking about protein or peptide chemistry, we often call these peptide bonds. So, if I have an amino group in one molecule, it can react with the carboxylic acid group of a second amino acid group in a condensation reaction. Remembering what a condensation reaction is, I have a carboxylic acid group reacting with an amine group to form an amide in water. Also notice here, I've drawn all of these molecules as if they are Zwitter ions, because that is the most common state for amino acids in living organisms in the neutral environment. This peptide bond formation is catalyzed by an enzyme that exists within the organelle called a ribosome within a cell. In other words, if I look at the reaction between a carboxylic acid group on one amino acid with the amino group of a second amino acid, that reaction is catalyzed within the ribosome within a cell. When those two functional groups react, they form a peptide bond, which we used to call an amide. We're now going to call it a peptide. I now have two amino acid residues. That is called the dipeptide. If I have a third amino acid, and it is catalyzed within the ribosome of a cell to form three amino acids. Here's one alpha carbon. Here's two alpha carbons. Here's three alpha carbons. I have two peptide bonds now, two amide linkages. We call this a tripeptide. If I continue this process, we call that larger molecule a polypeptide, which is the origins or the beginnings of making a long polymer protein. So what is the difference between a peptide and a protein? If my reaction contains two amino acid residues, we call that a dipeptide. If it contains greater than 10 amino acid residues, we typically call it a polypeptide, but less than 40. A protein is the term we use to identify a molecule which has 42 over 10,000 amino residues in it. If we look at a peptide, every peptide or protein has directionality to it. In other words, when I finally make my peptide or protein, I have an N-terminal end, which contains my NH3 plus N to it, and I have a C-terminal end, which contains my COO minus N. And this is directionality. In this molecule, I have the amino acid residue glycine, alanine, and serine, where I have peptide bonds between these three different amino acids. Again, here are the R groups hydrogen, CH3, and CH2OH. If we want to name polypeptides, we can just list those amino acid residues starting at the N-terminus end and going to the C-terminus end. In this example here, there are three amino acid residues. I have tyrosine, serine, and phenylalanine. I could list those, tyrosine, serine, phenylalanine, that would identify this polypeptide. I could abbreviate those amino acid residues as TYR, SER, and PHE, separated by a hyphen. Or if I want to even get simpler, I could just identify the one-letter abbreviation for each of these amino acid residue, Y for tyrosine, S for serine, and F for phenylalanine. IUPAC, however, has come up with a methodology where I name the parent chain as the amino acid residue that is near the C-terminus end of the polypeptide. In this case, that would be phenylalanine. And then I look at the other amino acid residues starting at the N-terminus end, and if the amino acid has an "-ine" or an "-ic" acid on it, I remove that from the name of the amino acid and add YL, sort of making it look like a substituent or an alkyl group. There is one exception to that. If my amino acid is tryptophan, then I typically just add YL to the word tryptophan. So this molecule here would be tyrosyl, serine, phenylalanine. This is my parent chain. That's the amino acid residue at the C-terminal end. And I start off here at the N-terminal end. Tyrosine, remove the INE, add YL. Then serine, remove the INE, add YL. Let's now do a practice exercise where we provide the IUPAC name for each of the following peptides. The first thing we do is identify the individual amino acid residues that make up the polypeptide. So I go between my carbonyl group and my nitrogen group. This is one amino acid residue. Here's my second amino acid residue, and here's my third amino acid residue. I know that because I can identify my alpha carbon in each one of these cases here. If I look at the names for these amino acid residues, that's alanine, leucine, and glycine, abbreviated either ALA-LEU-GYL or A-L-G. That's a very common methodology for naming this. If I look at the IUPAC rules, I'm going to name my C-terminal end as my parent compound, glycine. I'm going to then go start from my N-terminal end, and this would take off the INE. This would be alanyl, leucyl, glycine. If we look at this second example again, we're going to identify our amino acid residues. Here's one alpha carbon. Here's my second alpha carbon. Here's my third alpha carbon. So I have a tripeptide again. We then need to identify our side chain groups here, which will allow us to identify the amino acids. This happens to be glycine, alanine, leucine, abbreviated GYL-ALA-LEU or GAL for very short. From the IUPAC standpoint, we're going to name leucine as our parent compound and name glycine and alanine as substituents. So this will be glycyl, alanyl, leucine. If I look at this polypeptide, notice I've already just written it down as the three-letter acronyms or the single-letter acronyms, V, W, T, P. I need to identify those amino acid residues. They would be valine, tryptophan, threonine, and proline. Proline here would be my C-terminal end, because that's always on the right-hand side. That's going to be my parent compound. Each of these are going to now be my substituents starting off at the N-terminal ends. So that would be valanyl, tryptophanil, and threonyl proline. Because we give unique names to unique peptides, and because peptides have directionality, depending on which amino acid residue is the N-terminal amino acid and which is the C-terminal amino acid, they could be differences. For example, here I have alanine and glycine, where alanine is my N-terminal amino acid and glycine is my C-terminal amino acids. That's different than a polypeptide where I have the glycine amino acid residue on the N-terminal end and my alanine amino acid residue on my C-terminal end. If we look at the number of possible polypeptides, if I just look at the 20 standard amino acids and the possibilities of them, I could take 20 to the nth. Odds are that if I had an amino acid that has just 51 amino acids in it, there are over 1 in 2.2 times 10 to the 66, a very large number, to actually get them in the right order. They are put in the right order in living organisms by using the DNA and RNA of the cell. A number of the small peptides in humans function as messenger molecules, in other words, hormones, much like some of the lipids functioning as messenger molecules. Two of the best known small peptide hormones are oxytocin, which functions during uterine contraction and lactation during pregnancy, vasopressin, which regulates some of the kidney functions and also regulates some of the aspects of blood pressure. Each of these two small peptides contains 9 amino acid residues, so we refer to these as a nonapeptide. Notice that in the structure of this nonapeptide, amino acid residue number 1 is a cysteine molecule which contains a sulfhydryl, and amino acid residue number 6 is also a cysteine amino acid residue. These two sulfhydryl groups on there can form a disulfide bridge, folding it into this ring structure here and forming a small molecule that can be easily transported through the circulatory system and function as a messenger hormone. Both of these are produced by the pituitary gland, which is a small gland just off the front side of the brain here. There are a number of small peptides that function as neurotransmitters in the brain. One group of them is identified as enkephalins. They are pentapeptides which are produced by the brain, and they bind to receptors within the brain. An example of that would be if I have an interneuron, it has enkephalins in it. When they are stimulated, they release those enkephalins, which are then sent over to a sensory neuron, which stimulates them to actually produce a chemical reaction to sort of send signals to another interneuron, in this case to the pain centers in the molecule. Some of the best known enkephalins are tyrosine, glycine, glycine, phenylalanine, methionine, or tyrosine, glycine, glycine, phenylalanine, leucine. Those are slightly different pentapeptides acting as neurotransmitters within the brain. Let's now move on and talk about proteins. A protein is a polypeptide that typically contains 40 or more amino acid residues. The proteins are typically classified either by their structure, their shape, or their function, and we'll discuss that in detail. The terms polypeptide and protein are often interchangeable, meaning that one population might refer to a certain group of amino acid residues as a protein. Others might discuss them as a polypeptide. It depends on which type of scientist you are. There are several proteins that have more than 10,000 amino acid residues in them. The most common proteins contain somewhere between 400 and 500 amino acid residues. Other proteins may contain more than just one polypeptide chain. For example, if I have just one polypeptide chain, we call that protein a monomeric protein because it contains one polypeptide chain. If I have a protein that contains several polypeptide chains, 2 or more, we call that a multimeric protein chain, as represented here, where I have 1, 2, 3, 4, 5 different individual polypeptide chains which have come together and are held together by intermolecular forces to form a protein molecule or a protein structure. One methodology that scientists use to classify proteins is based on the structure of the protein. If my protein contains only amino acid residues, we call that a simple protein. That protein can either be monomeric or multimeric. This is an example of a simple protein. It's just a long continuous chain of amino acid residues. A second class based on structure are the conjugated or complex proteins. That is a protein that has one or more non-amino acid entity attached to it. We call these non-amino acid entities prosthetic groups. These prosthetics groups may either be organic molecules or inorganic molecules. If that prosthetic group, which is attached to my protein, is a lipid, we call it a lipoprotein. Here is an example of a very large lipoprotein. Sort of the green squiggly lines here represents the polypeptide chain. The orange molecules represent phospholipids. The blue dots on the outside here represent free cholesterol lipids. We also in the side here have triglycerides and some cholesterol esters in here in the middle. A lipoprotein. A glycoprotein contains carbohydrate groups or sugars. If I look at this molecule down here, the blue sort of spheres here represent an amino acid chain, which I have attached to it sugar molecules. In this case, I have a disaccharide attached to it, and in some cases I have polysaccharides attached to it. Glyco represents the word for sugar, and I have a glycoprotein. The third subcategory of conjugated or complex proteins are the metalloproteins, where my protein molecule contains one or more specific metal ions. In this example, I have a zinc ion that's here that's conjugated into this system and is held into this protein both geometrically and also by ionic forces. One way of actually classifying a simple protein is by their primary structure. The primary structure of a protein refers to the sequence of amino acids starting from the N-terminus end and going all the way to the C-terminus end. So every protein has a unique amino acid sequence. For example, here if I look at this polypeptide or protein, I start off here with glycine at the N-terminus end, then leucine, serine, and keep on going till I get all the way down here to the C-terminus end where I have glycine. That list of amino acid residue is used to define what type of protein I have. That's the primary structure of a protein, is the list of amino acids starting at the N-terminus end, going all the way over to the C-terminus end. The very first protein to actually be fully sequenced was by Fred Sanger in 1953. He was actually able to determine the amino acid sequence of bovine insulin starting at the N-terminus end, identifying all the amino acids to the C-terminus end. He also determined that bovine insulin is a multimeric protein. It contains 2 polypeptide chains. He received a Nobel Prize for this work. He received a second Nobel Prize for determining the methodology for DNA sequencing. We often identify the primary structure of a small protein or a polypeptide by listing the amino acid residues in the order they appear, starting at the N-terminus end down to the C-terminus end. It looks like a linear change of these amino acid residues. However, the bonding nature of the peptide bond actually gives it some geometric constraints to being linear. So if I look at the peptide bond in an amino acid, I have a nitrogen here with a lone pair of electrons. If I move some of that electron density between the carbon and nitrogen, it looks like I have a double bond now between the carbon and nitrogen. When I do that, I push some of the electron density away from the carbon-oxygen double bond to form a single carbon-oxygen bond. These two species are in resonance with each other. In other words, all of these atoms here will be in the same plane. If I look at the structure of a peptide bond in a polypeptide or a protein, I have my alpha carbon of one amino acid residue, my carbonyl group of that amino acid residue, my nitrogen of a second amino acid residue, and my alpha carbon of a second amino acid residue. These 6 atoms will be in the same plane based on the fact that they are in resonance with each other. In other words, I have a double bond that's between the carbon and nitrogen some of the time, and I have a double bond between the carbon and oxygen. That's going to put all of these atoms here in the same plane. What that gives rise to when I put that all together in a long peptide chain or in a protein is this trans-zigzag-like pattern. Each of these purple squares here represents a plane that is defined by 6 different atoms that would all be in that plane. Notice here I have 2 alpha carbons in this 6 carbon plane. Here I have an alpha carbon and an alpha carbon, my amino acid nitrogen and my amino acid carbonyl group. There's another plane, and etc. I have in this polypeptide chain here, I have 4 planar regions. What that also does, because of that geometry, it leaves this carbonyl oxygen out here that can hydrogen bond, and it leaves this hydrogen down here attached to the nitrogen, which can hydrogen bond. That has ramifications for additional structures. Let's look at that now. In addition to the primary structure of a protein, proteins have a secondary structure. This secondary structure is a spatial arrangement of atoms of the polypeptide backbone that is actually stabilized by hydrogen bonding between 2 different parts of that polypeptide chain. For example, if I look at one polypeptide chain, I could have my carbonyl oxygen opposite to the hydrogen on the nitrogen of a second polypeptide chain. They're going to form a hydrogen bond between the two and actually stabilize this planar geometry. In addition, we can look further down the chain. Everywhere I have a carbonyl on one of my polypeptide chains, I can actually hydrogen bond to my hydrogen on the nitrogen of a second polypeptide chain. Notice that these are actually in opposite directions. One of them starts at the N-terminal end is at the top here, where in this chain it's just the opposite. This is the C-terminal end. If I combine this sort of hydrogen bonding with the rigidity of that 6-atom planar structure of the molecule, I can actually form a 3-dimensional structure of proteins, either in an alpha helix or in what is called a beta pleated sheet. Let's look at that 3-dimensional structure now. The 3-dimensional structure of a polypeptide can also self-assemble into an alpha helix that is held together by hydrogen bonding. So for example, if I look at the carbonyl group of one amino acid residue, it can be in the right geometry to form a hydrogen bond with my hydrogen that's on my nitrogen of another amino acid group. If the geometry is correct, I can actually form this alpha helix here. If we look at this and we look at it from the top view, I can see that all my side chains are actually oriented outward. So all my R groups that are attached to my amino acid residues are sticking out. And I can look down on this and it sort of spirals down clockwise. That means it's an alpha helix. Another form of secondary structures that protein chains self-assemble into are beta pleated sheets. Again, these secondary structures are held together by intermolecular hydrogen bonds between strands of protein. So if I look here at this cartoon drawing here, this is a strand of protein made out of amino acids. It comes out here and loops around, and it comes back again like this. Notice here the red dots, which are the carbonyl oxygen on one part of a strand, they line up with the hydrogen attached to the nitrogen on a different part of that strand, and the geometries make what is known as a beta pleated sheet. An example of a beta pleated sheet and how it is represented typically is shown here. Again, a long strand of amino acids making up a protein. They're in this beta pleated sheet, sort of this zigzag configuration. An example of that would be the proteins that make up silk. And this sort of a soft, springy type of protein, which exhibits itself in the silk when we touch it. Most proteins have a combination of different secondary structures. Some portions of a protein may have an alpha helix secondary structure, which are shown here in these helixes in this specific protein. Other section of that same protein might have beta pleated sheet regions, the secondary structure shown here. And there's also going to be some unstructured sections of that protein chain. Usually, the unstructured sections sort of connect the alpha helix sections to the beta pleated sheet sections. These unstructured sections are also much more flexible than the alpha helixes or the beta pleated sheets. In addition to a primary structure and a secondary structure, there is also a tertiary structure of a protein, and that gives it its overall three-dimensional shape. This three-dimensional tertiary structure is a result of the interaction between atoms that are widely separated in regions of the primary structure. So one end of the protein chain might be attracted to another end of the protein chain. So if I look at an example of that, you can see there are alpha helixes, and these ones are held together, close together, to give it this whole structure of the protein here. This is some unstructured protein sections. So both the backbone of the atoms and the side chains of the atoms can participate in giving a protein this overall three-dimensional tertiary structure. Let's look at some of those forces. So if I look at the tertiary structure, we're typically looking at the interactions between R groups. For example, in one section of my protein, I might have a cysteine amino acid. Somewhere else on that long polypeptide chain, I might have another cysteine amino acids. Both of those contain the sulfhydryl groups. Those can actually form together to be oxidized to form a disulfide bridge. That holds these two sections of the protein together. I also have electrostatic regions of my protein chain. This is due to the fact that part of my protein chain is a zwitterion. So I'm going to have negative charges and I'm going to have positive charges. Those attract each other. So here is one portion of my protein chain. Here's a second section. They are attracted to each other. We often call this a salt bridge because oppositely charged side chains are either acidic or basic, and they are attracted to each other. And then we also have some side chains, some R groups, that can hydrogen bond. For example, in serine, I have a hydroxyl group. In glycine, I have this amide group. The hydrogen on my serine is attracted and hydrogen bonds to the oxygen of my glycine molecule. And the fourth type of interactions are my London dispersion forces, just nonpolar ends that set up a temporary dipole and they track to each other. So these four different forces hold that tertiary structure together. Even though many of the individual forces are weak, there are so many forces that it is actually net a strong force holding a protein in this tertiary three-dimensional structure. Let's look at a simplified diagram of a protein and those four different intermolecular forces that hold that protein in that tertiary three-dimensional structure. In this diagram, these red spheres here represent individual amino acids that have a peptide bond between them to make up a long protein chain. This particular amino acid has a hydroxyl R group on it, and this particular amino acid has a carboxylic acid functional group on it. These ones have the right type of chemistry to hydrogen bond and hold this part of the peptide chain close to this part. There are also interactions with London dispersion forces. These are forces between hydrophobic groups. So this amino acid is attracted to this amino acid, again, helping make that three-dimensional structure a little stronger. If I look at this amino acid, that would be a cysteine amino acid, and this amino acid is also a cysteine amino acid. They have a sulfhydryl group, which can be oxidized to form a disulfide link, holding this part of the peptide chain close to this part of the peptide chain. And finally, we have these salt bridges, where I have ionic compounds at normal pHs within living organisms, where I have an anion on one amino acid and a cation R group on another. These are attracted to each other. All of these forces and many, many more of the similar forces hold this peptide chain, this protein chain, together in a tertiary structure. It is important to keep in mind that many of my side groups, the R groups on my amino acids, also have amines and carboxylate groups, and therefore they will exist as a Zwitter ion. And so my protein itself is going to be charged at different places around the protein depending on what those side chain groups are. And if I go into basic solutions, I'm going to end up with an anion. If I go into acidic groups, I'm going to end up with cation charges on it. And they're going to be attracted to other anions, or in this case, the anion is going to be attracted to positive anions. So these proteins can actually be attracted to each other. The charge distribution on the side groups is responsible in part to organize proteins into larger groups, which we call multimeric proteins. In other words, we have more than one protein chain coming together to form a greater three-dimensional structure. In this case, we call it the quaternary structure, and it has to include at least two or more polypeptide chains coming together. This is the highest level of protein organization, and excluding the possibility of disulfide bonds between these different protein chains, these subunits are held together by hydrogen bonds, by London dispersion forces, and by these salt bridges due to charge distribution. Let's now review the 4 different levels of a protein structure, starting with the primary structure. So the primary structure is the sequence of amino acids present in a protein's peptide chain, or in this diagram, I see 2 chains here. So those amino acids all have in common an alpha carbon in which there is a carboxyl group attached to that carbon, an amino group attached to that carbon, a hydrogen attached to that carbon, and this side chain attached to it, represented here by R. There are typically just 20 different R groups that are present to make up 20 different key amino acids in living organisms like humans. All of these amino acids are the L isomer because that is a chiral carbon, meaning it's got 4 different things attached to it. So there is a D stereoisomer also, and that has to be synthetically made. If we look at those amino acids that come together, they react to form an amide bond between them, often referred to as a peptide bond or a peptide linkage. Here we have 1, 2, 3, 4, 5, 6 different amino acids with different R groups on them that have formed peptide bonds. If we start to name these, we always start naming a protein by starting at the N-terminal end, naming each amino acid residue until we get to the C-terminal end. That is shown here in a larger polypeptide chain or a protein. In this case, we represent each amino acid by a 3-letter acronym: CYS for cysteine, SER for serine, LEU for leucine, and PHE for phenylalanine. Also notice that these peptides and these proteins are defined by the type of amino acids that are present, the number that are present, and the order that they're in. That's the primary structure of a protein, often referred to as a polypeptide chain 2. The secondary structure in a protein is defined by the ability of the carbonyl group on part of the protein chain to hydrogen bond to the amino group on another part of the peptide chain. That is shown here in this diagram, where I have 6 atoms that all sit in the same plane. My carbonyl group can hydrogen bond, and the hydrogen on my nitrogen can hydrogen bond. What that does, it allows one part of a polypeptide chain to hydrogen bond to another polypeptide chain, either by the molecule wrapping around upon itself or by forming a helix type structure that leads to 2 different types of common secondary structures: the alpha helix and the beta pleated sheet. Again, these are held together in these 2 different structures by hydrogen bonding between that carbonyl oxygen and that nitrogen hydrogen. The tertiary structure of a protein is due to the interactions between different R groups on different parts of the polypeptide chain. They can either be due to disulfide bonds, electrostatic interactions, salt bridges, hydrogen bonding, or hydrophobic London dispersion forces. So let's look at those again. I could have hydrogen bonding between 2 different side chain groups on a polypeptide chain. I could have London dispersion forces. I could form disulfide bonds between different amino acids in different portions of the polypeptide chain. And I can finally have that ionic bond or salt bridge that holds this molecule into a 3-dimensional structure. If we look at this, typically there are regions within a molecule that contain alpha helixes. I can also find sections that have beta pleated sheets, and I could have unstructured regions Those are all held together in a three-dimensional geometry by these four different forces from the R groups, the side chain groups on the amino acids. And finally, we have some proteins that are organized into quaternary structures. We often call these multimeric proteins because they contain more than one individual primary structure. In this diagram here, I have four that are held together by electrostatic interactions, hydrogen bonding, or hydrophobic interactions of the R groups. Again, those interactions, I have my salt bridges, I have my hydrogen bond, and I have my London dispersion forces, all leading to some three-dimensional structure, in this case of four different protein chains held together. Proteins undergo chemical reactions. One of the most common chemical reactions that proteins undergo are hydrolysis reactions. That's where I take a long polypeptide chain, I react it with water and some catalyst to actually break that peptide bond. The resulting materials are my carboxylic acid group and my amine group. If these are on a protein, I'm going to have two new freed amino acids. Effectively, we've just reversed the polypeptide bond formation process. Digestion is an example of an enzyme catalyst hydrolysis reaction. So in living organisms, with an enzyme, I can start breaking up my polypeptide chain into individual amino acids. Those freed amino acids are absorbed and transported to the liver, where they are used to synthesize new proteins. So hydrolysis of cellular proteins and reutilization of these amino acids is a continuous process in the body. Let's look at an example. Here I have a tripeptide of alanine, glycine, and cysteine. If I add water, in this case we'll use an acid catalyzed reaction and some heat, I'm going to break these two polypeptide bonds, add water across them to form three individual amino acids. So are these amino acids zwitterions? Well, in this case, no, because I'm under acidic conditions. If I were in a living organism and I used a catalyst, these would be in a zwitterion, or they'd be ionic based on where they are in the living organism. Before I can actually do hydrolysis reactions, I need to actually first break up the secondary, tertiary, and quaternary structure. Let's look at the processes to break those up. Proteins have a very organized structure to them. This ordered three-dimensional structure is due to disulfide bonds, electrostatic interactions, hydrogen bonding, and my London dispersion forces. If we break up that three-dimensional organized structure, we call that denaturation. And if I look at that, what I'm essentially doing is I need to break all those intermolecular forces that hold that protein in that three-dimensional structure. Some of the common ways to do that are to change the pH, to raise the temperature, or change the solubility by adding some sort of chemical reagent to it. So if I take an ordered protein, I denature it using one of these three or some other processes, it loses all its biological activity. It no longer functions as a protein. It sort of unwinds, if you will, because again, we have none of these forces holding it together. In living organisms, I can put them back together into this three-dimensional organized structure. We call that renaturation. Let's look at some specific examples of protein denaturation. One of them is actually the coagulation that occurs when we cook an egg. Eggs are very high in protein. When we cook an egg, we're actually changing the protein structure. In this case, the alpha helix becomes unbound it unwraps, and so I have an unstructured protein, and it is no longer soluble in water. So an egg, before we cook it, is soluble in water, and when we cook it, the white part also becomes insoluble. So cooking denatures the protein. By doing that, it actually aids us in protein hydrolysis, so it's easier for us to digest. Also, because microorganisms also contain protein, we also denature their protein, which kills them. And so we're actually sort of protecting ourselves from those proteins, those microorganisms, so that we don't get sick by cooking our food. Our body also has a way of denaturing proteins when we become sick. Oftentimes when we are sick, we have a fever, sometimes over 104 degrees. What happens in that case is some of the proteins in those microorganisms or bacteria, they start to denature. In other words, their three-dimensional shape changes. We're going to find out in the next chapter that many proteins are actually enzymes. They're catalysts for chemical reactions. If we denature those proteins, those enzymes, they change their shape and they can no longer catalyze reactions. So if a bacteria can no longer catalyze chemical reaction, it dies. So it's a defense mechanism by the human body to have a fever. Another example is getting a permanent. If you have curly hair, we have a lot of keratin proteins, which has a lot of alpha helixes in it. When we have a permanent, we actually chemically change the nature of those proteins to straighten that hair. That's because curly hair has a lot of disulfide bonds between the different protein strands. Chemically, we can break those bonds and straighten our hair. Let's now look how scientists classify proteins. One methodology is to classify proteins based on their three-dimensional shape, and we'll start off by discussing that one. The second way that scientists classify proteins are based on its function. So if we look at shape, we can classify proteins as either being fibrous proteins, meaning the molecules are long in shape. These tend to be insoluble in water and tend to aggregate to form macromolecular structures like hair, fingernails, and toenails, things like that. Usually they have a single type of secondary structure, meaning they're all either alpha helixes or all beta-pleated sheets. The second type of protein classification is globular proteins. That's proteins that sort of form in these sort of compact, sort of spherical type shapes. They're often water soluble because they have hydrophobic residues that are buried inside here. But they have a very diverse functions. They usually act as enzymes, or they can act as intracellular signaling molecules. And finally, membrane proteins is another class, and they're always associated with membranes. If we look at a bilipid layer made out of fatty acids, we need some way of getting materials into the cell or out of the cell. It turns out that proteins act as a conduit to help transfer molecules across that membrane. These are typically insoluble and unstable in water, meaning that they like to be in that membrane. The second methodology that scientists use to classify proteins is based on their function, and we find out that proteins are actually a very versatile molecule in the body, and they play very crucial roles in most biological processes. So proteins are diverse in function, and they exhibit functions that far exceed the role of any other biochemical molecule. In fact, one of the goals of living organisms is to produce proteins that can actually help the organism survive. So that functional versatility of protein stems from three different types of phenomena. One is the ability to selectively and strongly bind to small molecules. You can see here in some of these examples of proteins, you can see other things just sort of sitting down in there that are bound to the proteins. They also have the ability to interact with other proteins and form fiber-like structures. For example, in this example here, I have several different proteins here binding together, and some of them are going to be fiber-like. And also is the ability to integrate into cell membranes and actually act as transport conduits for different materials. Let's now take a look at the functions, the classes of functions that scientists have put proteins into. Let's look at the 12 different categories used to classify proteins based on function. Number one is catalytic, enzymes catalyzing reactions within living organisms. Defense, they bind to foreign substances to combat invasion, like an immune system. They transport, in other words, they bind to small molecules and transport them to other locations in the body. There are messenger proteins that transport signals to coordinate other biochemical processes between the different cells, tissues, and organs in the body. One of the most important ones are the contractile proteins. In other words, those are the ones that are used for our muscles so that we can move. Structural proteins, those include our tendons, ligaments, vessels, and skin. Transmembrane proteins, they span a cell membrane and help regulate the movement of small molecules and ions in and out of the cells. Number 8 here is our storage proteins. They bind to and store small molecules so that when our body needs them, for example, a vitamin or a mineral, it can release those into our system. Regulatory proteins, they act as sites for receptors on enzymes and control their function, so they regulate some of the enzymes. Nutritional proteins actually function as nutritional sources. That's especially true in early life, like in milk proteins. Some proteins act as buffers to maintain our acid-base balance so that pH in our body is always maintained at a constant level. And finally, the 12th category is fluid balance. That means we need to maintain the pressure between our blood vessels and the surrounding tissue in order for those water molecules and those water soluble molecules to be transported out of the bloodstream and into the cells. Let's now look at each one of these 12 functional classes of proteins in a little more detail. Number 1, catalytic proteins. Catalytic proteins are enzymes. They catalyze most biochemical reactions within living organisms. An example of that would be this globular protein here. This protein is actually sucrase. It takes a molecule of sucrose, which is a glucose and fructose molecule. That molecule fits down into the three-dimensional structure of this globular protein. It puts on stress here between the glucose and fructose molecule and facilitates the hydrolysis of that molecule into individual glucose and fructose molecules. Again, this is a catalytic protein so that another sucrose molecule can come down fit down into the molecule and undergo a catalytic reaction. This stress in here between these two different molecules of sucrose and fructose lowers the activation energy for that chemical reaction. That's the definition of a catalyst. A second class of proteins based on function are the defense proteins. These are part of the immune system in living organisms. They're often referred to as immunoglobulins because they are globular proteins by nature. The general public often refers to these as antibodies. They bind to foreign substances in an effort to combat invasion. of those foreign substances. They are multimeric proteins in nature, meaning they have more than one polypeptide chain, 4 in this instance. Immunoglobulins are also glycoproteins, meaning they have an amino acid section of the protein, but they also have a sugar part of the protein. If we look at this sort of cartoon drawing of an immunoglobulin or antibody, the yellow things here would be antigens or the invasive species. The protein binds to that antigen, and then it allows it to be carried away through our bloodstream and into our excretory system here to be eliminated as waste, and therefore removing that antigen from the body. A third class of proteins based on function are the transport proteins. They bind to small biomolecules like oxygen and transport them to other locations in the body. A common example is hemoglobin. Hemoglobin is a protein that binds to oxygen in our blood cells, and it transports oxygen from our lungs to tissues throughout our body. Those are typically globular in nature. They're multimeric. In the case of hemoglobin, it contains 4 different polypeptide chains. It's usually a metalloprotein also. In the case of hemoglobin, it contains an iron molecule, and it contains 4 of them. Each subunit contains one heme group, and it allows the hemoglobin to transport up to 4 oxygen molecules at once. So oxygen from the lungs comes in and binds to the hemoglobin in a red blood cell. It gets transported down the circulatory system to different tissues. When it reaches a certain point, the proteins release that oxygen into the cells, and that oxygen is used for metabolism. A fourth category of proteins based on function are the messenger proteins. They transmit signals to coordinate biochemical processes between different cells and tissues and organs in the body. A couple examples of that are human growth hormone, which is a multimeric globular protein, and it regulates body growth. Insulin is another messenger protein which regulates carbohydrate metabolism. So insulin lowers the blood glucose by enabling glucose metabolism within cells. Insulin is a multimeric protein also. It contains 2 different polypeptide chains connected together by some disulfide bridges. Diabetes is a disease characterized by high levels of sugar in the body, and it's the insulin level that helps us regulate that sugar. Therefore, insulin and diabetes are closely related. Let's take a closer look at insulin and diabetes relationship. So in people that do not have the disease of diabetes, glucose stays at a healthy range. That's because we have the correct amount of insulin, represented by these triangles here in this cartoon. They actually let that glucose molecules, represented by these cubes here, into cells where they can undergo metabolism. And it's the insulin that helps the glucose enter through the cell membrane into the cell. We often refer to those who have diabetes as having a high blood glucose level. In other words, they're hyperglycemic. Glycemic again meaning sugar levels. There are 2 types of diabetes. One is that if we have too little insulin is made by our pancreas, we don't have enough insulin to help move that glucose into the cells, or the cells can't use insulin very well, and that's type 2 diabetes. If one looks around the world, you can see a different prevalence for diabetes. This is an estimate of what the diabetes levels will be based on populations by the year 2025. You can see here in the United States and Canada, about 8 to 10% of the population has diabetes. There are certain regions of the world where we have greater than 20% of the population has high diabetes occurrences. And there are other parts of the world, for example, here in Asia and in Africa, we have very low levels of diabetes. That comes from both diet and from genetics of that population in those areas. A fifth type of protein based on function are the contractile proteins, in other words, our muscles. There are two types of proteins that help us to actually move. Those are actin and myosin. These are fibrous type proteins and they are multimeric proteins. You will learn a lot more about this in physiology classes of biology. The very rudimentary explanation is that we have one protein, the myosin here, and actually they are held together by these Z discs here, and then when there's signals sent to the muscles electronically, they tend to work sort of like Velcro. They contract the muscle to actually allow us to move. A sixth type of protein based on function are the structural proteins. These actually provide stiffness and rigidity to the body. Collagen is one of those structural proteins, and it is the most abundant protein in humans, making up around 30% of the total body protein. They are fibrous and multimeric in nature, and they tend to be in this triple helix here. They are the major component of our tendons, our ligaments, our blood vessels, and our skin. They are also the organic component of both our bone and teeth. The primary structure is rich in the amino acid proline, and you can actually go to the drugstore and buy collagen protein to help with sore muscles and help to loosen up some of the skin molecules and make it more smooth, if you will. Another important structural protein is alpha keratin. It is the major protein component of hair, feathers, fingernails, horns, and turtle shells. It also provides protective coating around many of our organs. It's mainly made out of hydrophobic amino acid residues, meaning nonpolar alkanes, and they're usually in a alpha helix secondary structure. In other words, they're coiled. The hardness of a keratin, whether it's a hair or fingernails or horns, depends on the number of disulfide bonds. The more disulfide bonds in a keratin protein, that makes it more look like hard nails, or makes our bones hard, or makes our hair even brittle. So when we look at horns or we look at hair, we tend to think of alpha keratin as that protein making up those structures. They're fibrous in nature and they're multimeric in nature. A seventh class of proteins based on their function are the transmembrane proteins. These proteins span the cell membranes and help control movement of small molecules and ions into the cell and out of the cell. They come in two different varieties. One are channel proteins that just have a channel through them, so it's a three-dimensional shape. It lets through anything that's the right size. So here is our bilipid layer cell membrane. Here is the protein that spans that membrane. A second type of protein are carrier proteins. These have a shape that actively transports only certain molecules in. So here we have a set of molecules, and if they fit directly into the protein, and we need more of them in this case on this side of the protein, it will open up and let it through. This is just a drawing of a cell and a cell membrane. Notice here we have proteins spanning that cell membrane. An eighth class of proteins based on function are the storage proteins. These proteins bind to and store small molecules that might be needed in times of stress, or they might be needed as we change our diets. Two examples of those are ferritin, which is a globular monomeric metalloprotein. It actually stores iron and saves it for use in the biosynthesis of new hemoglobin molecules, which are the transport proteins used to transport oxygen around the body on our red blood cells. Another important protein related to ferritin and hemoglobin are the myoglobin proteins. They store oxygen and they store them in our muscle cells. So when a muscle is active and it needs oxygen in times of stress or strong exercise, that protein will release that oxygen from its protein, so it serves as a reserve energy source for working muscles. Myoglobin. A ninth class of protein based on function are the regulatory proteins. These proteins are effective in telling a living organism to start making a chemical or stop making a chemical, and they often do that by changing an enzyme. We know that an enzyme promotes most chemical reactions in a living organism. If I need more of a chemical compound made, I have an enzyme here with a very specific shape, but it's the wrong shape to do chemistry. I have a regulatory protein that binds to my enzyme, changing the shape so it's just the right size in order to attract those substrate molecules and start doing chemistry. If I have too much of a molecule, I can actually stop chemical reactions by, again, changing the shape of an enzyme so that it can no longer act as a catalyst for a chemical reaction. I'm regulating the production of chemical compounds by changing the shape of enzymes, and those are done by very specific proteins in the body. Most of them are globular in nature, and they're also multimeric in nature. A 10th class of protein based on function are the nutritional proteins. Nutritional proteins are an easy source of amino acids, and they're usually very important in early stages of life, from all the way from the embryo through infancy. An example of a nutritional protein is the protein in milk, casein. Because there are no disulfide bridges in casein, they're relatively easy to hydrolyze. They also have a property that they have an ability to form a gel and clot in the stomach, which makes them very efficient as a nutrient supply. Another example of a nutritional proteins are egg whites. Even though the function is not entirely known by scientists, it is assumed that they are a good nutritional source of proteins. Most eggs have a lot of this egg white protein in them, and we know that that's what the developing chicks or developing animals use as their nutritional source until they hatch. An 11th class of protein based on function are the buffer proteins. These are the proteins that help maintain the correct acid-base balance in the body. One way they do that is the fact that proteins have both an amino group and a carboxylate group on them in the form of a Zwitter ion, either in the R group or in amino acids. If I have a Zwitter ion and I'm in an acidic medium, I can actually act to absorb some of that hydrogen acid and form the cation here. If I have a situation where I am in an alkaline medium and I want to neutralize some of that alkaline medium, I can actually pull a proton off of the amino group, thus removing some of the hydroxide ions. In addition, buffer proteins can also regulate ion flow across membranes. In other words, these are channel proteins that also function as buffer proteins, letting ions through or preventing some bigger ions from going through from the outside of the cell to the inside, or vice versa, from the inside to the outside, thus maintaining the correct acid-base balance within the body, within the cell. A 12th class of proteins based on function are the fluid balance proteins. These proteins help maintain a fluid balance, water balance, between the blood and the surrounding tissue. Two proteins, albumin and globulin, counteract the loss of water associated with the heart pumping. In other words, blood is being pumped through our bodies at high pressure. Proteins in the blood attract that water and keep them from diffusing out of our blood vessels and our blood veins. In other words, they balance the fluid level within both the circulatory system and within the cells. These are typically globular proteins, so they cannot go through the cell membranes that are associated with the veins and arteries in a body. And that ends our discussion on proteins. Next, we're going to move on and talk about both enzymes and vitamins.